So next, we're moving into a little bit of drug delivery. So we're going to hear about EyeDura Therapeutics from Srini Venkatesh.
Thank you. Good afternoon. I'm Srini Venkatesh, CEO of EyeDura Therapeutics, and it's a pleasure to present at Eyecelerator. Okay. So if you look at neurotrophic keratitis, it's a rare disease that results in the innervation of the cornea. And if left untreated, it can lead up to vision loss. So this is something that we are approaching at EyeDura. The current standard of care for neurotrophic keratitis is Oxervate. That's the drug manufactured by Dompe Pharmaceuticals. And the drug works. It's a nerve growth factor. It's an 8-week intensive therapy. And despite its clinical efficacy, Oxervate has certain challenges with respect to the frequency of dosing. You need 6 times a day for 8 weeks. And you can think of that from a weekly perspective of 42 drops per week. And the patients have to go through a 19-step process for preparation of the dosing solution. So in this context, we want to introduce EyeDura, where we are transforming the delivery of topical eye drops. EyeDura was founded to address this critical challenge of balancing efficacy of the drug along with compliance and ease of use. We have a proven board, a strong leadership team, and a group of advisors with deep experience in ophthalmology drug development, commercialization, and fundraising. So what is this technology? We have a novel mechanism of action. The basic insight we have is that when there is a blink, the aqueous layer is selectively removed much faster than the lipid layer. So if we had a formulation that resides on the lipid layer, then it can form a stable depot on the eye and provide durability. So that is the key insight. Our formulation is ID002, which is human insulin eye drops. The formulation is based on a lipid vehicle, and it has a high viscosity profile. And as a result of this, our value proposition is that we have reduced frequency of dosing, we have simplified administration from a bottle or from a single dose unit, and improve patient compliance. And you can have once-a-day dosing. Here's an animation that I'd like to share with you.
Applied in the same manner as conventional eye drops. On the corneal surface, the TSR formulation binds with the lipid layer. Because the lipid layer does not drain quickly, this creates a long-lasting depot of medication on the surface of the eye. Over multiple days following the installation of a TSR eye drop, much of the active ingredient will make its way into the eye, enabling a more efficient use of pharmaceutical ingredients and a longer-lasting effect than conventional eye drops.
We have a strong patent portfolio that takes us up to 2044 with 8 issued patents and 10 published patents that you can see, and they're globally. We ran a study in Central America where we dosed 10 patients with persistent epithelial defects, and we dosed them, and these patients were refractory to all the treatments that were available, and we dosed them with our insulin eye drops. After 1 to 2 drops per week treatment with the EyeDura drops, the epithelial defects heal completely in 10 out of 10 cases. So this is a remarkable finding and needs further exploration in the clinic to run systematic trials both for safety and efficacy, and that's exactly what we're going to do at EyeDura. We have recently announced a partnership with Singapore Eye Research Institute to use their mouse model for preclinical efficacy to test our insulin formulation with the EyeDura vehicle. The EyeDura platform can go well beyond neurotrophic keratitis in terms of going into dry eye and neuroprotection for glaucoma patients. In fact, recently we ran a study in Honduras with 17 subjects where we used a probing trial, of course, with our ID001 ophthalmic solution formulation. And we used the OSDI scores, that was licensed from AbbVie. And we showed that there is a strong trend in improving the ocular surface index score in these 17 subjects over a 4-month trial. So these are very encouraging. And finally, we also tested our insulin drop in Dr. Adriana DiPaolo's lab at the University of Montreal, and we demonstrated that in our GC cells, we can provide neuronal survival. So we are excited to demonstrate these data. In closing, we want to thank you, and we appreciate your attention here.
Thank you.